Today we'll be learning about frameshift mutations. This is the table we'll use. It's called the codon table. It lets us translate the three bases into the appropriate amino acids. Notice how AUG for methionine is coded in green for the start codon, and we have a couple stop codons in red. So first of all, we have to separate them into three bases. That's why they're also called triplets. So we have AUG for methionine, that's M. We have GCC for alanine. And then we have AUU for isoleucine. We have AUC for isoleucine again. And then we have GCU, which is for alanine. And then we have CUU, which is for leucine. And we have AGC for serine. And we have UAA, which is a stop codon. Now that's how you would normally read this amino acid peptide, polypeptide, but there's a few ways we can change that through mutations. This arrow was transferred a little incorrectly, but bear with me here. First, we will learn about silent mutations. We'll split the frames again. So if we translate this arrow indicates on the third base of the second triplet of codons, the A is replacing the C. Now it's AUG from methionine, the start codon, and GCA instead. GCA also codes for alanine, and everything else stays the same. GCU and stop codon. The silent mutation is also known as a synonymous mutation. It means which are usually third base substitutions, which does nothing to change the function of the amino acid. Now let's look at some non-synonymous mutations. In this case, we are changing the first base of the second triplets from a G to an A. We'll start off with methionine again. Now, if you remember previously, that was a GCC, which codes for alanine. Now, since the first base changed from G to A, it will be ACC, which is threonine. And then it's isoleucine, GCU, alanine, leucine, serine, stop codon. So instead of MAIIALS, we now have this threonine instead of alanine. It changes the amino acid function, which makes it a missense mutation. Now we will look at this. This is an insertion of G in between those two. We'll start off with methionine, which is a start codon, GCC, which is alanine, AUU, which is isoleucine, and look, now we have UGA. UGA is actually a stop codon, which means this amino acid right here stops. And all this after does not matter because it stopped. This is known as a knockout mutation. It cuts off the amino acid chain very suddenly. Lastly, we have deletion. This U that's underlined is now deleted. You have methionine, the start codon, GCC for alanine. Now you have the U that's deleted, so you still have an AUA, which is isoleucine. I have UCG, which is serine, CUC, which is leucine, UUA, leucine, GCU, alanine, and something else. It could either be asparagine or lysine. Really depends on the third base here. But that's how you can tell there are various mutations and how they could happen. And they're all called frameshift mutations.